Good evening. My name is Leslie Behar, and I'm a proud Technion supporter from the San Francisco Bay Area and a member of the ATS Silicon Valley Board of Directors. In fact, my mother, Elaine Ehrman, was a founding local board member, and we have a long legacy of family involvement around the country. I'm delighted to be with you today to introduce you to someone who is not only a shining example of Technion ingenuity, but also a close personal friend, Professor Adam de Lazerda. Adam is a Technion alumnus, graduating in 2005 with a bachelor's degree in computer engineering. Received his PhD from Stanford in electrical engineering and conducted postdoctoral studies at University of California, Berkeley. He is now an associate professor in the Department of Structural Biology and Electrical Engineering at Stanford University School of Medicine, and the founder and CEO of Visby Medical, makers of a revolutionary, portable, single-use PCR testing platform that can test for infectious diseases such as COVID-19, as well as others. This device, this platform, holds the potential for opening up our lives to travel, concerts, school, theater, and all the things we've been missing for so long. Adam has been featured on CBS News, CNBC, and on the TED Talk stage. As a friend of the De Lazerdas, I'm so proud of Adam's success. As an investor in Visby, I'm delighted that the hard work Adam and his team have put in over the last several years is coming to fruition and receiving the recognition that it so greatly deserves. On a personal note, I met Adam about 15 years ago when he was a first-year grad student at Stanford. We met through his then girlfriend, now wife, the amazing Adi Solomon de Lazerda. Adi was about to start her Technion studies, but was visiting in the Bay Area for the summer. We bonded over our shared connection to Technion. Adi finished Technion with a BS in chemical engineering and is now a Stanford PhD in materials engineering. We've become close friends, almost family. As amazing as Adam and Adi are academically and professionally, their true accomplishments are the three cutest, smartest, smiliest kids you'll ever meet: Noah, Yair, and Ido. Now, before we begin, just a reminder to please use the Q&A function on this webinar screen to submit your questions for Adam, and we'll get to as many questions as we can at the end of the program. I'm honored to welcome my dear friend, Professor Adam de Lazerda, to the screen. Adam, welcome.
Thank you so much, Leslie, for the very, very kind introduction. And thank you to the Technion ATS team for the kind invitation here. So Technion has been a very important part of my life. Obviously, first and foremost, this is where I met my wife, Adi. This is also my alma mater, where I really got my most important basis for my educational career. And really, I think for those of you that have gone through the journey of the Technion, I think you'll appreciate that. An expression that comes to mind is what doesn't kill you makes you stronger, is very much appropriate here, because it really is shaping an individual on so many different levels. The whole point about personal career growth through education, through challenging ourselves personally and professionally is obviously through learning how to deal with stress, with a lot of things happening at the same time, juggling a lot of things, and really still keeping the bar very, very high throughout that process. And certainly Technion has been just gets an A+ on being able to provide this kind of environment for its students. Many other things too, but that's maybe one of the things that comes to mind. What I'd love to share with you today is a story that has to do with infectious diseases. Now, before you dismiss it by saying, well, infectious diseases isn't this thing that only third world countries deal with, which is still important, but not here in the United States or in Israel or in Europe somewhere. The answer is absolutely no. Obviously, many of you know COVID is an infectious disease, actually, and it's an infectious disease that is caused by the coronavirus, a particular type of strain of coronavirus that just emerged, obviously, a little more than a year ago now, and completely changed our lives. And it's just important to recognize that while COVID is obviously huge now, and hopefully, and we have all reasons to be hopeful that it will titer down over time, and we can talk about it more later, and if you want in the Q&A session as well. The world of infectious diseases is still very much here, and it's not going up and down because of COVID, but it's just fundamentally viruses, bacteria, parasites, fungi have evolved over billions of years, way before we were here, to adapt to new changes, to opportunistically try to create new kinds of strains. And then once something kind of works well, in the same way biology works, survival of the fittest, start thriving. And if it bumps a couple of people along the way, then so be it, because that's how biology works. And so the world of infectious diseases is absolutely fascinating on so many different levels. Obviously, we're being affected by infectious diseases every winter when we get either the flu or the common cold. If you're a young child or an elderly person, or maybe just somebody with some pre-existing condition, other viruses, respiratory viruses such as RSV, respiratory syncytial virus, could actually get you pretty badly. My own daughter, Noah, that Leslie was mentioning, when she was 1.5 years old, was hit pretty badly with that virus, which led to just a terrible, terrible process that we had in the hospital at Stanford, which fortunately she made it through, but it was just very, very scary few moments there. The world of infectious diseases, of course, extend way outside of just respiratory viruses. This is where we think about just any place in the body that we humans can get infections, from eye infections to ear infections to skin infections to urinary tract infections, sexually transmitted infections, and yes, also pandemics such as COVID and others. And so there's just a lot to talk about and unpack in this story. What I'd love to tell you tonight as kind of a main story is share with you a little bit about what the company that I'm involved with has done in the context of infectious diseases. Most importantly, what are we trying to do? And really try to get to the heart of what are some of the important things for humanity to solve and for all of us to be cognizant of as we continue our lives, hopefully way past COVID. Obviously very much of an interesting topic today, but hopefully one that will stay with us for many years beyond that, because it's just a lot of things we need to do. And I think we've all seen what is the impact of not being ready for a pandemic that's coming, which we were all, as nations, were illfully unprepared for what has come upon us here with COVID. So that is the main story tonight. And I thought it would be maybe will be fun to maybe share with just a couple of things just prior to that, just tell you how I've gotten to even work on infectious diseases, because truly nothing in my background is qualifying me supposedly to work on infectious diseases, or frankly biology or medicine for that matter. At Technion, I studied computer engineering, which is kind of a mishmash between electrical engineering and computer science. And just to keep things interesting, I started spending quite a bit of time in the physics department there. I really finished the Technion with a very, very clear-minded idea that I'm going to be working on quantum computers, which is a really cool, sexy topic that at least I found to be really interesting about 15 years ago now when I finished. And because of that, when it came time to apply for graduate school, Stanford was just a very clear choice, because Stanford happened to have had one of the best quantum physics groups, quantum computer groups in the world. So that was a clear place to end up in. So I got in here, and through a pretty unfortunate, or very unfortunate circumstances, that completely changed immediately when I arrived here. A very good friend of mine, Ehud Ravitz, which studied at the Technion with me in computer science, was just a brilliant kid. The kind of kid that just always gets an A+ at the Technion. It's like somewhere between 98, 99, and 100. If you got anything under 98, it must have been a really bad day for him. just one of the most brilliant students. He obviously finished first in his class, and as he started his army training right thereafter, he started failing all sorts of exams. And when they try to understand why is it that Ehud is not doing very well, they realized that unfortunately Ehud had a brain tumor. It was already a stage 4, and Ehud really did not have a lot of chances here. And unfortunately, within about a year or so, he passed away. And that precisely coincided with the time that I started my studies here. And it really struck us pretty hard. None of us were prepared for that. We all felt that the medical community would be able to help him. He was always incredibly positive. If you knew Ehud, you would know that he was just always the most optimistic person. He could always conquer anything and everything. And clearly that did not happen. And several of us, his friends and myself included, that really struck us pretty badly. And we said, well, we have to change what we're doing, how we've been thinking about our purpose in life. For me, that came really at an interesting time of starting my graduate studies here, and very quickly decided what I really wanted to do is to go into medicine. Of course, at the time, I didn't really know what's the role of an engineer like me in medicine, and through some interesting journey, eventually realized that actually it turns out engineers do have a role to play in medicine and biology, which might be completely obvious to all of you. It was not at all obvious to me at the time. And I ended up finding myself really starting that journey now 15 years back, and really have not looked back since. The interesting thing about having, as you know, an academic career, and for me, I kind of juggled back and forth between Stanford and Berkeley and back to Stanford in the medical school, is that if we're successful, we would turn really good ideas into papers where we will teach the world about whatever we invented. But there was something that felt a little dissatisfying, but just stopping there. Obviously, you cannot treat a patient, you cannot diagnose a disease with a paper. You need to turn that into an actual practice, into a product, into a process. And living in Silicon Valley with a lot of VCs all around us, I ended up joining this venture firm here in Palo Alto as an entrepreneur in residence. I have to admit, at the time, the intent was not to start a company, but rather really to learn more about how companies get started. And through this process of maybe spending about 2 years with them, the idea was for us to create an investment hypothesis, something that we would say, like, if somebody can really solve this problem, it'll be a very worthy problem to solve, and we should invest in them. So I spent about 2 years doing that, looked at about 50 some companies. It took that long, but finally, after about 2 years, it occurred to me that I might actually know how to solve this problem. And so I went back to the VC guys and said, guys, we're not investing in anybody else so far, at least. And so I'm starting a company. If you guys want, you're welcome to invest. And fortunately, they did, and that's where we got started. And that was really the basis for Visby Medical. I'll tell you a moment more about what we're doing, but we've had a pretty interesting journey here. So we started a company about 8 years ago. We grew quite a bit during that time. We raised about $300 million across several different rounds of investments. And also the team itself here grew quite a bit. About a year ago, when the pandemic just started in March, we were about 90 people or so in the company. Now, fast forward about a year, we grew from about 90 people to now a little over 700 people, all of us based here in Northern California. So it's been a pretty dizzying year on many different levels. And just managing that kind of growth is something that, if any of you are interested, happy to talk about it more in the Q&A session. But maybe it's a good place to just start by telling about what do we actually do, what are we actually solving for. So as we would all expect, the way that medicine is being practiced, just about in any major field of medicine, is we would first want to diagnose somebody, and then we will treat them based on the diagnosis, right? So we would first diagnose somebody with cancer, and only then would we consider giving them chemotherapy. We'll never do it the other way around, of course. Heart diseases, we first diagnose somebody with heart disease, only then we do an open heart surgery. Diabetes, you first diagnose that, only then you give the person insulin. There's only one major field of medicine where we're actually not doing that. That works completely backwards. And that's the world of infections. where we are still first treating you and only then diagnosing you like a week later. And this is the fundamental problem that we're trying to solve. Not only it's a $200 billion market that works backwards, there are a lot of lives of people behind it that are greatly impacted, because when it comes time to make a decision on how to treat this patient, we just don't know what this patient has. Not only do we not know if this is bacterial or viral infection, which in and of itself is a big deal, but we would love to know what kind of virus, what kind of bacterium. If it is a bacterial infection, like a urinary tract infection or something, or respiratory infection, we really want to know, is it resistant to a certain antibiotic or not? So we know what the right antibiotics to give to that person. These are all really important questions. And at the time when you, the patient, is sitting with the doctor in that urgent care center, you truly do not know what you're dealing with. And the doctor doesn't know what you have. You know the doctor doesn't know what you have. The doctor knows that he doesn't know what you have. And so there's a lot of mistrust in this whole system. And that's one of the fundamental problems that we would really love to try to solve. So let's double click on that problem a little bit. Because you see, the thing with infectious diseases is that when it comes to diagnosing infections, it is actually not about coming up with a new technology. There is already an existing technology that works exceptionally well. The technology, like Leslie mentioned before, is called PCR, stands for polymerase chain reaction. I can talk about it more afterwards if you want what it actually does, but long story short, it's a big machine the size of a sofa or so. It comes with those cartridges. You typically would find it in a big laboratory or a hospital, maybe sometime at the basement of a hospital. And those big machines that companies like Roche and Abbott and so on make are very, very accurate. As in, you can take a sample. If for COVID, for example, it could be a nasal swab or saliva. If it's a different part of the body, you just swab that particular part of the body you're worried about for infection, you send it over to the lab. The lab runs that test. It's actually a genetic test. It can really look for the individual genetic code of the pathogen. So it's exceptionally accurate. If it finds that genetic code, it means that you have this virus or bacteria, and there's no way around that. And so that big PCR machine finds out what it is that you have, and then the doctor gets back to you. Of course, by then it's been at least a few days, if not a week. And obviously, we had to make a decision on how to treat you before that event happened. So that is the fundamental thing we need to try to solve. It's not about reinventing PCR to whatever the next generation technology is. It's really about solving the market access problem of the existing technology. How can we make PCR accessible to anyone? And anyone truly means anyone. And that was that investment hypothesis we started from. Can we take that big PCR machine and make it accessible to anyone? So big sofa-sized instrument. What we said, at least at the time, was we really want to go for, we need to go for something that look and feel like a pregnancy test. It cannot be a big instrument, not even a small instrument. It needs to be something that you go to CVS store, you buy it, you run it, you throw it away, or you order it online or something. It needs to be incredibly accessible. It needs to be super easy to use, and surely it cannot take many hours to run. It needs to run relatively quickly. So the question there was really, can we take a big instrument that comes with those cartridges, a razor blade model, and can we fuse the instrument and the cartridge into the same unit and package it so that the whole thing is a single-use product, and ideally one that doesn't cost you $1 million, because then you're not going to use it very often if that's the case. So that's where we really got started, and we've been working on it for several years, and fast forward a few years, we did it, and it actually works rather well. Actually, I brought it here to show you. So the device I'm holding here, hopefully you guys can see the video feed, is the world's first and only single-use PCR machine. Even though it doesn't look quite like one, it is actually an entire PCR machine. It's a little hard to see, that's the whole point. It's kind of small, but let's see if I can bring it a little closer to the camera. There we go. So here you can see our first product. This one is the one that's looking for sexually transmitted infections. You'll notice that it has an opening right here. This is where the sample goes. So if you're a doctor and you have a patient in front of you, instead of collecting that sample and sending it out to the lab, you collect that same sample, but you now put it here. For females, for example, it could be a vaginal swab. And once you put the sample here, running it is not that hard. All you have to do is just close this lid like so. And now you see there are these 3 buttons here. You just have to press them in that order, 1, 2, 3, and in less than 30 minutes, you'll get an answer right here. Now, in this case, we're looking for those 3 sexually transmitted infections, chlamydia, gonorrhea, and trichomonas. The exact details of them matter less, but it's one of the most common panels that people are running, at least in the United States, with about 50 million tests like this every year. And in this particular case, within minutes, you can see whether the patient has this, that, or the other. What matters here is those 3 infections we're looking for there all have identical symptoms, but very different antibiotic you need to treat to it. Going back to the question before, don't just tell me it's a bacterial infection. I already know it's bacterial in that particular case. I need to know which one of them is it. So I know if I should give you this antibiotic or that antibiotic, or maybe both, because sometimes people have more than just one infection at the same time. And so that's really the fundamental technology. And it's been quite a journey to develop that, about 60 people working for quite a few years. And then we found ourselves in February of last year. We just finished a clinical trial for this device that I just showed you. Almost 2,000 patients across 15 different sites in the United States. We're kind of going head to head trying to compare our small little device with the big PCR machines. The clinical study results came back and it looked really good. Basically, we're able to show that essentially they're performing at the same level. Somewhat to be expected, it really is the same technology. And so at that point, we submitted that, of course, to the FDA, and they've started reviewing that. They're still reviewing it, by the way. But while they've been reviewing this, we've been asking ourselves, well, this is more than just a product; it's a platform. By taking this physical device and just exchanging the chemistry here, by just telling this device what genetic code we want to look for, we can turn it from a device that's looking for sexually transmitted infections to devices looking for any other virus or bacteria you care about: eye infections, ear infections, respiratory infections, you name it. So we're going through these product strategy meetings, thinking through, well, what should be our next target. And of course, Q1 of last year doesn't take a genius to say, COVID is pretty big, and maybe COVID should be our second product here. And so by March, we were already full force working on this. This is what I mentioned to you before. We really looked at this and said, all right, the technology seems to work well. It really is a platform. And this is going to be far bigger than any of us can even anticipate how big. None of us can really anticipate how big it is, but we all can agree it's going to be huge. And so let's start working on a COVID product. So we got into this hiring frenzy mode, if you will, starting to really grow the entire team, more than just an R&D team now. Now we need to start thinking about, it's not just about designing and developing it, we also need to manufacture this, and ideally in high volumes, because the world's going to need that. And not only manufacturing this in high volumes, we should also make sure that it's not just the assembly process, but the entire supply chain that has been completely crushed by COVID all over the world. We need to really rethink that. So we had to rebuild we had to build from scratch a lot of new areas that were completely foreign for us, that we never even thought we would have to get to so quickly, and start this journey with COVID. Didn't take us 6 years like the first product. It took us only 6 months this time to develop the test that I'm holding here, which you'll notice looks very similar to the first device I showed you. If this one is the device that we developed for sexually transmitted infections, this is the one we developed for COVID. And you'll see that this one also has this opening, but instead of putting a urogenital sample, you put obviously a respiratory sample. So you swab the nose and you put it in here. Close it, you press the 3 buttons, and again, less than 30 minutes, you get an answer here, kind of like a pregnancy test. 1 line means you're negative, 2 lines means you are positive. And now we are finding ourselves in September of last year with a device that works well, finished all the development, clinical studies, submitted that to the FDA. Now, FDA doesn't take 12 months like our first product. In this case, FDA is working around the clock, literally, and giving us the authorization in 2.5 weeks. So kind of shows you that when they really want to move fast and everyone is completely incentivized, they can really move mountains. And so FDA in their heroic work have been moving incredibly fast in this. And we find ourselves in the market, which was also an interesting journey. So there's a new company just putting our first product out, trying to figure out, well, we have this product, we're now in the height of the pandemic, where do we deploy it? The unique thing of what this thing can do is it can give you not just an accurate answer because it's a laboratory quality equipment. It's an actual PCR machine in your hand, but it's also really quick. It's all very rapid, and it's completely portable. So in theory, at least in the future, the idea is that anyone will be able to look for any infection, anywhere, anytime, whatever context you might be in. In the case of COVID, there are so many different cases and locations where people want to get tested. So obviously we start first by prioritizing the frontline healthcare workers, those vulnerable patients that are maybe either sitting in a cancer center, in a nursing home, dialysis centers, and so on. So we start really there. And we start working with all sorts of laboratories as well that want to start buying those tests. These are big laboratories that are serving large communities. A lot of people are sending samples over to them. And this is where things get really interesting, because we're seeing that there's this interesting thing that people told me that many years ago, that when you launch your product, you think you know what people are going to do with it, but then once you launch it on the market, you really see what people end up doing with it, and it could be very different than what you thought they would and how they would use it. This was completely the case here. There are all these labs that start buying our product, and initially we go like, what the heck are you doing with our product? This is a big lab. You have all these big, humongous PCR machines the size of a sofa. Why do you need to buy this small little PCR machine? Well, it turns out the people behind those labs are very creative business people. And they're not just excellent laboratorians and clinicians, but they really try to think about what do the end user want? What do they need? And they realize that they can actually take our physical product and turn it into a service product. And some of them even go all the way to actually build an entire VSB van, as they call it. There's several of them running around different places. And those VSB vans are testing people in different places. So some of them have gone into schools, and they were able to, once they go into a school that was in a full-blown outbreak mode, they were able to stop the outbreak by just running as many tests as needed at the same time on every single one of the kids and the teachers, and make sure that they can completely stratify immediately who's positive, who's negative, right there, right then. From there, it started to go into other interesting places like a wedding and so on. That was also, we were also able to have. Several of those were also used in Washington, D.C. during the inauguration, and several other interesting places that we never quite anticipated would be of interest for a device like this. But in retrospect, of course, makes a lot of sense. So that's a little bit kind of the journey that we've gone through. It has certainly not gone without massive amount of challenges, which I can tell you a little bit about now. And of course, if you're interested, I'll tell you more in the Q&A. But supply chain has perhaps been one of the most challenging things. You think that the hardest thing is to design a product that works, but that's not at all. That's only the first part of it. We have a lot of components that go into every one of these devices. It's a fairly complicated device because, again, it's taking the full big instrument and just miniaturizing it, but there's no shortcuts here. It's still the same machine, And there are a lot of different components, some really expensive ones and some pretty insignificant ones you would never think twice about. For example, we had this double-sided tape in this machine. It's a very simple component. It costs maybe a penny or something, less than a penny. There's this big company that sells to us this double-sided tape that needs to go in. And as it turns out, if you have all the components to build a device, but you do not have this double-sided tape, you have no device. You just cannot build it. So a single component not being available can completely stop and halt your manufacturing line, which actually happened to us with a couple of suppliers, including this one really annoying supplier that makes this double-sided tape for us, that just got to us at some point and said, sorry, we know we told you that we'll ship you X amount next week, but we're delayed and we'll ship it to you 3 months from now instead, which, as you can imagine, causing a lot of people here to be pretty worried. Now, this is also where you can really see where governments can really step in really nicely. And for those of us that have been in the diagnostic industry now for the last several months, you know that the US government, as well as other governments around the world, really stepped in and were really trying to help very quickly, to the point where the case of that one example I shared with you, people that are very high up in health and human services in HHS literally picked up the phone, called the CEO of this multi-billion-dollar company and said, Mr. Such and such, please take a pencil and a paper and write down purchase order from this small little company of Visby Medical. And we want to ask you to please, that strong please, make sure that this order gets fulfilled and by next week at the latest. And that actually helped. So literally by the following week, we actually did get the part that we needed. So there were a lot of interesting challenges like this, a lot of firefighting mode over this past year. And truly kind of resonating with the same thing I said about the Technion earlier, and now living it up a little bit later. What doesn't kill you makes you stronger. You put yourself in a really, really stressful situation, and you try to make the most out of it. And certainly that's something that every single one of the people that are sitting behind me here have really been experiencing. Let me tell you maybe one last thing before we open up for questions, which is you'll notice that I'm here in our headquarters, and I'll a little bit over. And if you can see, this is our main headquarters here in San Jose, Northern California. We were operating now, about 95% of us are back in the office, working from the office, which is not so typical these days. So maybe I'll share with you how that came to be. Obviously, one of the perks of being a manufacturer of COVID tests helps us create a COVID-free zone, but that also wasn't without some challenges. So in October of last year, we got to the point where we had a lot of people already in our manufacturing lines. It is impossible to keep social distancing. If you could work from home, you work from home. But if you're working in the labs or in the manufacturing rooms, there's just no way you can do that. And we're doing everything we possibly could, with the exception of testing, because at the time we still did not have that capability of being able to do that. And so we've been testing people on a regular basis, but of course, you collect the sample, you send it to the lab, and by the time you get the result, it's been a few days. And so our first COVID case showed up on a Tuesday. And of course, when that happens, we immediately do contact tracing. We look at all the immediate contacts. And then everyone that's in immediate contact, we immediately send it out for testing as well. And so by Thursday afternoon, we got our second COVID case. And that second COVID case was connected to the first COVID case. So if it's just one case, that could be an isolated case, but two cases are connected, it's a full-blown outbreak. So at that point, which is Thursday afternoon, we immediately shut down the factory. We closed down the entire office, sent everybody home. We brought in all the senior management, so the senior vice presidents, the vice presidents here. And we worked all through the weekend, and we said, come hell or high water, Monday morning, we have to get back to operation. We are going to test every single person in this company before they make it into this office. And we actually did. We found a way to do that. I got my senior vice president of supply chain watching the door, being the doorman. I got my other senior leaders to collect swabs from people with N95 masks and so on. And that Monday morning, we tested every single person that morning. We found 23 people that had the virus, and fortunately, everyone are doing well by now. And later that day, we were able to get back to operation. And since then, we've been running this experiment, which we call Visby at Visby, which of course under medical supervision and so on, to really allow us to operate in a way that at least we feel that's safe. And that's a pretty remarkable story also, when you have to, for us here, when you have to respond very quickly. Obviously, we're considered an essential industry, and we want to very much stay in business and be able to continue to produce those tests. And the only way to do it is we can, if and only if we can keep our employees here safe. And that's how we're keeping that. Anyways, I have plenty more things I'm happy to tell you. If any of you are interested, happy to talk more about variants and how does that affect. It's always an interesting topic and very much relevant now. But let's maybe stop here and pass over back to you, Leslie, and open up for questions.
All right, thank you, Adam, for that fascinating look into your own journey and into that of Visby. Before we move on to the questions, and there are a number of them, I thought I'd get a chance to ask you a couple of my own. So you've mentioned a couple times about what you learned, how Technion prepared you. Just wondered if you wanted to tell a story about your time at Technion.
Oh, I've actually quite a few. So being a little bit of a geek, perhaps I'll share that it was quite often that I made it to professors' rooms to either ask them questions or ask them more questions, or maybe argue about a grade or something. I know if you have one incident, maybe you can blame somebody else for that. If you have two incidents, maybe it's still a coincidence. If you have three incidents of something that's kind of awkward, maybe it's you. So I have the honor, which I'm assuming is probably the only person, but perhaps, I don't know, maybe there are others. I have the only honor of having 3 badges of times when an instructor or when a professor at the Technion literally told me to get out of their room because I'm just outright annoying. And by the way, very likely they were right. By the way, so I might have not recognized it at the time, but very likely they're probably right. But you know, like, just I think one of the most incredible things that I remember from the thing, and maybe that one thing aside, which is a completely true case, by the way, but that one thing aside, I think one of the most remarkable things I remember from the Technion is just how everyone just very much internalized that we're all in this together, and we're all working together, and this is incredibly hard journey that we're going through right now. And just the degree of collaboration, you know, in a way that might be hard to explain to people that have never gone to university in Israel, perhaps. So like here, you know, when I'm teaching in class at Stanford, everyone knows that we're curving the grades, and if you get an A, then that means that maybe I'm getting a B because it's curved. Technion, that we never thought like this. At the Technion, we're all in this together. We're helping each other. We're studying together. We spent enormous of time together, and it was just such a beautiful community there. Of really just incredibly high caliber people. So one of the best friendships I've had and still kept to this very day are my friends at Technion.
Thank you. That's heartwarming. I love to hear positive experiences and warm fuzzies from there, even that I don't like whoever told you were annoying. That's not okay.
No,
okay, I wanted to move on to some of the questions that have come in through our Q&A. I'm going to get to some of them. I don't know if I'll have a chance to get to all of them. But let's start with Angela is asking, is this device a one-time use, or does it come with the swabs in the kit? Can it be administered by anyone?
Yeah, thanks for the question, Angela. So the device is a single-use device, a little bit like a pregnancy test, very much intended to move away from the concept of an instrument and a cartridge, especially because in this case, we're dealing with an infectious sample. And every time you're loading up an infectious sample on an instrument, you have to worry about cleaning the darn thing after you're done. And if you're not careful, you could also cross-contaminate. So one really positive sample could now contaminate several other negative samples, which we've seen actually happening now during the pandemic, especially in the case of COVID, where sometimes these nasal swabs can come back with literally billions and billions of copies of virus in that one swab. And now if there's a small amount of that virus that crosses over to the next run, all of a sudden you're getting a false positive. So yes, it is a single-use device. One thing that may be worthwhile mentioning is when you're done running the device, we're actually setting up a recycling program. We're a little bit like those Nespresso cups. When you're done with it, you can actually send it back to us. And we're working this reverse logistic process now, especially for those larger customers of ours, just to make sure that this thing, as much as possible, at least gets recycled, and in many cases even reused. That's something we're working on right now. Now you're asking, does it come with the swabs in the kit? So I should maybe mention that we're initially focusing this particular device on the hospital, healthcare setting, and point of need markets. This is not yet sold at home for people to be used in their, in their own home. Obviously, because it's a single-use test, this is where we're ultimately heading. Our long-term vision is to be able to make this available to people at home, not just for COVID, but literally for any infection you care about. Other people might care about sexually transmitted infections. We might care about respiratory infections, broadly speaking, like flu and others for young kids or elderly parents. So we are trying to head to the at-home use. And when we do that, the device will come with everything you need, including the swab itself. But for now, we're focusing on those other settings, which technically are called the CLIA labs and the CLIA wave settings, which now means schools, kindergartens, universities, cancer centers, dialysis centers, urgent care centers, airports, many corporations like us want to get back to work, and all these kind of larger settings where you want to try to create either a COVID-free zone, or you want to really make sure you can do testing and tracing well. So instead of coming to somebody and said, hey, you've been exposed to COVID, go and get tested, we can now say, hey, you've been exposed to COVID, here's a test, let's test you right here, right now. And if you're positive, let's test your immediate contacts and their immediate contacts until we get the entirety of the infection chain and stop it there.
All right, I'm looking, we got a whole lot of questions. So I may pick and choose, and if your question doesn't get answered, I think somebody's gonna save them and pass them on. Where are these tests manufactured, and how many tests have you manufactured?
A great question. So we are manufacturing these tests right here in Northern California. We have suppliers all over the country that are making the individual components for us. But you can actually, if you look back, you can actually see the two large manufacturing rooms. This green room that you can see right behind some of the screens. And right here, there's a white room over there. So this is where we're actually manufacturing the tests. We're now at a production level of tens of thousands of devices per month. Not nearly enough. Obviously, we want to try to grow a lot faster than that. We're investing a lot of efforts and have been receiving very large grants from the federal government here to help us scale up the production, mostly through automation. And we're making some pretty substantial investments there. So we're hoping to get to hundreds of thousands and beyond that pretty soon.
Here's one for people who've been reading the news. Is Theranos still having an effect on getting VC funding for diagnostic enterprises?
That's a really interesting question. So for the very few of you that don't know what Theranos is, you really would enjoy reading an amazing book called Bad Blood. And because of the sake of time, I'm not going to take you through what's actually there, but you'll really enjoy reading it. I promise you, it's not science fiction, even though as you read through the book, you at some point are left to wonder, is this really science fiction? But for those of you that are not familiar with that, it is perhaps one of the largest fraud schemes that have happened in the world of medicine, really in modern history. Now, Theranos had a pretty substantial effect on funding to, especially the diagnostic industry where Theranos was operating for a number of years, had a huge impact both on their way up and on their way down. And we actually very much felt it both on the way up and on the way down. On the way up, Theranos basically made the claim that you can test for just about everything with a small little drop of blood. And so in that setting, it was a little challenging to get VC money to really support that, because many VCs said, hey, there's already another company that can do just about everything. And of course, on the way down, many VCs realized, hey, maybe we should not be involved in this whole business. But I would say the important thing is that the investment community really recovered from that and very quickly realized that diagnostics is a very important industry. If you look at the overall healthcare spending in the United States, diagnostics is maybe about 5% of that. But if you ask yourself, well, how much of the healthcare spending is directly impacted by diagnostics, decided on by a diagnostic test? It's about 85% of it. So it has a huge leverage to it. If you make a good diagnosis or a bad diagnosis, you impact not just a lot of patient lives, but also enormous amounts of dollars. And so for all the right reasons, this is, I think, a very interesting industry. And I would think, actually one of the things we're seeing now just over the last year is almost some form of a renaissance, of course, on the heels of COVID, which is very unfortunate, but some sort of renaissance for the diagnostic industry, that across the board, even in the world of cancer diagnostics and others, cardiac disease diagnostics, a lot of companies are now kind of having an opportunity to really pull up and start to be more recognized, which is wonderful because it's a recognition that this industry can really benefit from and the world can really benefit from.
I'm sure you get a lot of questions about the Theranos impact.
Sure.
Let's see. Again, I apologize if I'm skipping some of these. Is there a timestamp on the test to show when the test was taken, as in when proof is needed of a negative test for a specific time frame, like for flights or stadiums or other venues?
Yeah, it's a great question. So think about our test a little bit the same way you would use either rapid test like a pregnancy test. In and of itself, it doesn't have a timestamp. It's just a test that you're running on you're running it and you're getting a test result. Now, when run, for example, by airports or by some deployment partners of ours and so on, then it's really that deployment partner, maybe at a school, that it is really on them to generate that report for you, whether that's a text message that's sent to you, or maybe like a connection to a digital passport, which there's so many today, where whoever ran the test, which initially is really in the professional settings, and can now connect to your mobile phone in some way and give you that kind of stamp of approval. But once it does happen, what you get there, and whether that's through any one of those digital passports that exist today, it will tell you that you were tested using a PCR technology, that you were tested hopefully negative, and where that took place. So this is all part of the ecosystem that we're now part of.
Oh, cool. What happens when the virus mutates just slightly or more than that?
Yeah. So first of all, maybe let's just recognize the fact that viruses mutating is not a new thing. It's not necessarily something we have anything to do with. That's just what they do. Viruses, bacteria mutate. By the way, human cells mutate too. That's what created evolution. That's what created differences among people. And viruses certainly mutate. This novel coronavirus was created through mutations of an older version of a different coronavirus. And over time, it mutated enough through different species that it resulted in something like this. It is very much survival of the fittest. For every millions and millions of mutations that take place, there might be one that is slightly stronger, more infectious perhaps than the other ones. And through survival of the fittest, that's the one that's now having an opportunity to continue to mutate off of that. And so really the question is, what is really driving those mutations? Why are they bad? And really what should we be anticipating to see over the next several months with respect to those kinds of mutations? Maybe the best way to think about it is you can think about the timeline from the day the pandemic started, and you can really try to identify 3 different regimes of times. The first one, really the wild type. This is really the virus that has not mutated yet. This is just the virus not mutated, going into a population that's never been exposed to it, doesn't have any immunity to it, and really taking off very quickly, unfortunately, as we know all happen. So that's really the first phase. Now, the second phase is where we're at right now. In that second phase, you have naturally occurring mutations. This is just random mutations that the virus picks up. Most of these mutations are bad for the virus or not helpful, so they kind of die off very quickly. But every once in a while, you have a mutation that really helps the virus spread more. The South African mutant, the UK mutant, now the Brazil mutant, the Californian mutant, and so on. These are all naturally occurring mutants. And the selection there is for a virus that's slightly better at replicating, infecting a little bit more, and so on. That's the second phase we're at right now. The third phase is the phase that's called the selective pressure phase. The selective pressure phase is really the big unknown, the big question here. If any of you have heard Tony Fauci talking to Congress a few days ago, you know that he's been talking quite a bit about the risk for not just the existing mutations, the existing variants that were created naturally, but for selective pressure, a true genetic pressure that's applied on the virus from any host of things, whether those are antibodies generated due to a vaccine, or whether these are antibodies generated because somebody already was exposed to COVID before, and now the body develops some degree of immunity. And so in that kind of environment, mutations might try to select for being, again, survival of the fittest, being able to thrive under that immunity environment. This is not different than the way bacteria mutate when you throw a lot of antibiotics at them. And as we know, is the more antibiotics you throw at them, those mutations that happen at the bacterial level will cause antibiotic resistance. Well, viruses mutate the same way, sometimes even more. And that's really the third phase. Now, none of us really know how big that third phase could potentially be. There's a lot of estimation around this, but really it's really on us to really find out. What we do know is the more vaccination we're applying, the better we'll be, because there'll be less opportunities for those mutations. We'll try to bring down the levels of replication cycles and opportunities to create mutants as much as possible. And the more testing and tracing we do, the more we can hone in and limit the number of opportunities for the virus to replicate, the less random mutations we will get, and the less genetic pressure we will have later on in that phase. But for that, we need to apply all these techniques, not just vaccines, but also testing and tracing, not just in one geographical area, but across the entire world. And that's one of the reasons why in the world of infectious diseases, having an outbreak of any sort anywhere in the world is bad, because ultimately we're all very much connected here. And it just takes one passenger to fly from one place to another to bring that virus or bacteria over. And now you have a full-blown outbreak in the other area as well.
We're getting short on time. I'm going to give you a 3-part question. How robust is the platform? What are the false positives and false negatives? And has it been tested in large numbers in different environments? And how does it compare to other devices that are being reported on as being similar, also small and portable?
Yeah.
Sorry, that's a lot.
No, that's a really good question. So hopefully I can tackle at least most of them. So when it comes to the robustness of a platform, the important thing is what is the underlying technology you're running there. And in this particular case, we just found a way to take the big PCR machines and miniaturize them. But point being, if you're a virus and I put you into one of these devices, you would never be able to tell the difference between this device and a big machine. It would truly feel the same for you. And it's really because of that when we ran our clinical studies, we saw that the level of accuracy, whether that's the sensitivity or the specificity, are very much similar. And that's, again, to be expected, whether you're looking for this bacterium or that kind of virus, it doesn't really matter. At the end of the day, PCR is PCR is PCR. And so from that point of view, we feel that it's fairly robust. One of the interesting things about it is it's robust across multiple different diseases as well. So the fact that we've done some work earlier on a fairly large clinical study with thousands of participants on the sexually transmitted infection test gave us a lot of comfort, and ultimately, the regulatory bodies such as the FDA, to say, well, now all you're doing is really taking the exact same physical device, you're switching out the chemistries there just to tell this device what genetic code you're looking for. Now, the important things when you're designing this, going back to your later question about false positives, false negatives, the important thing is to make sure you're designing it to look for the genetic code of the pathogen. That is what's called the conserved region of the pathogen. And this is where there's a lot of bioinformatics work. You want to make sure that in all these regions where the virus can pick up all sorts of mutants and all the variants are created, you want to stay very far away from that. Because as those mutate, you might start missing things. So you might have the spike protein. The spike protein is really that thing that gives coronavirus its name from the crowns. It's those proteins that allow the virus to dock into a human cell and really create the infectiousness. This is where all the action happens with respect to mutations. So we're trying to stay very far away from that in an area that's not mutating as fast. And from a bioinformatics point of view, a very boring area of the genetic code of the virus, and specifically look for that. So now you're asking, like, how is it different than other systems out there? So generally, when you think about testing, there's so many different options for testing. Broadly speaking, you have PCR and you have non-PCR tests. The non-PCR tests, for example, could be tests like antigen tests, and they're not necessarily bad. They're not at the same level as PCR. If you're looking for people that non-symptomatic people at the open population, you might miss a few people. And very much depends on the setting and how badly it is that you want to get the answer right. If you're going to visit maybe a loved one in a nursing home, you really want to be sure that you're not carrying something, and only 70% accuracy is just not acceptable. By the way, in a very similar way, we love to talk about sensitivity, but specificity is also really important. So we have quite a few nursing homes reaching out to us, telling us that the rapid tests that they're running, sometimes are giving them false positive tests. Now you would think, well, what's the big deal? False positive test, just quarantine the person for a few days and that's it. If you're in a nursing home, that's a big deal. Poor grandma gets a false positive test result. You're moving her over to the COVID ward. If she did not have COVID before, now she might get COVID from other people in that COVID ward. So for all the right reasons, we care about both sensitivity and specificity. Of course, differently at different levels in different settings. And this is what PCR can do exceptionally well, not just our test, but any other category any other test that's in the big PCR device. And of course, what we do well is we find a way to take the big PCR machines and just make them a single-use PCR device. So now you can just carry it anywhere with you.
Awesome. Thank you, Adam. There are lots more questions that we're not going to get to, but we will try to get your questions answered if they weren't. You check with the ATS folks on that. Sorry if I threw you guys under the bus. Thank you, Adam, for this fantastic conversation. You are such a good storyteller and explaining and bringing us along on this journey. Thank you. Your story is the embodiment of what we refer to as the Technion impact, both the high-tech sector as well as in medical advancement space. You make us proud to be supporters of the Technion, and your brilliance gives us hope for the future. Thank you to all of the viewers for joining us today. You will receive a copy of the recording of this program in the next few days. And in the meantime, I wish you good health, be safe, and sending the warmest wishes for a joyous Passover. Chag Sameach.
Chag Sameach. Thank you.